Hi, everyone. Um, so I'm going to go over a few things. First of all, we'll start with how to analyze your data, which you'll need for your lab report. Okay. So, I'll upload this in just a minute. This is our data. This is, well, sorry, it's actually combined between the two sections, just so we have a bigger sample size. So, the um, first thing we want to do is we want to convert these to percentages, right? Um, when it comes to any kind of data, recognition, problem solved, it's just easier to think of it in terms of um, percentages, right? Rather than raw numbers. So the way to do that is it's pretty easy. You're just going to type a little formula. You type equals and then whatever cell you want. Okay, so oops, should be A2. So just going to type equals A2 right over here. That's, as you can see, it highlights that cell. Now, that's just selecting the cell. So since we wanted to convert it to a percentage, we're going to divide it by the number of trials in that condition. So if you remember, there was 15 trials total that you could get right? 15 rat trials. 15 per condition. So to convert this first one, we're just going to type equals 82 divided by 15. So that's the number they got out of 15. Right? Well, it's not working. Oh, sorry. I think I had the slash go the wrong way. So just check that. Okay. And then we're just going to do the same thing for the control trial. Okay. We'll see where this is going in a minute. So type same thing, but it's going to be B2. So B2 right here. You can see it gets the right one. Divided by 15 again. Here we go. Now, the reason we do that is now we can just copy and paste it. And what Excel will do is it will update it. It's all based on relative position. So what will happen is if you copy this and paste it along here, it'll switch it from A. For this one, it'll be, A3, A4, and all that stuff. So if you need to analyze some data pretty quickly, this is a great way to do it. I don't know. Okay, so I'll just show you that again just in case. So copy this, and then you just paste it along here, along all the other values. You can do either paste or function, just not values. So there we go, formulas or functions. And there you can see that's everyone's score in percentages. So as you can see here, the left one is the incubated trials and the right one is the control trials. So the incubated trials, remember, that's where you got 2 10-second attempts. Control is where you just got 1 20-second attempts. Right? So let's also just have a look at our means. So formula for that, you're just going to type equals average. Oh, and just before I move on though, and just remember it's within subjects and there's only 2 conditions, right? So everyone just does incubation and control. The only reason we split people up is because we want to counterbalance, right? So half of you do the incubation trials first, half of you do the control trials first, right? To offset any fatigue or any practice fix, right? So moving on to the average, we're just going to type equals average, pretty straightforward. And then just in brackets here, you can either highlight or type the numbers you want. So I'll just highlight it. So you just go over it like this, oops, I right click there. Just highlight all these numbers, but don't highlight the cell you're typing this into. Enter, there we go. And then you can actually just copy and paste this one, okay? So as you can see here, there does seem to be an effect, right? On average, people got 56% of the incubated trials compared to only 47% of the control trials. So that's good. And that's what we predicted, right? The question is, of course, is that a significant difference, right? Is that beyond what we could just expect due to random fluctuation, error variance, stuff like that, right? So that's where we'll run our test. Okay, so what you're gonna do is you're gonna open up a site called, just give me a second. What the hell? No, I need this. Okay, all right, so you're just gonna go to a site called vasarstats.net. So that's VASSAR stats.net. Okay, so I'm gonna save kind of just a review of stats for the most part for another lecture, for the results lecture. For now, just kind of remember what we talked about in our research review. So for this one, it's just going to be t-tests and procedures, okay? And it's going to be 2 sample t-tests for independent or correlated samples, okay? Okay, so hopefully you're on this page. So we'll just break some of this down. So independent samples means between subjects, right? So when we do a test, we it runs a little bit differently depending on whether it was a within subjects design or a between subjects design. Okay, so since this was a within subjects design, we're going to click correlated samples. Okay, independent samples means between subjects design. Correlated samples means within subjects design. Okay, so what you're going to do is go back to Excel. I have to keep switching my screen, so just hang on. And grab all the data from this first column here, except the mean. So don't grab this last value that you created. Copy it, and then you're going to paste it in one of those boxes. Okay. So paste it in sample A here. Just remember which condition was which, and then do the same thing for the other column. Okay, so just scroll to the top, make sure you have all these numbers. Make sure this looks right to you. So that's what you have. If you need any help with this, just send me an email. Calculate. Okay, so we're not going to worry about most of this. All that's important is really this part here. So are these couple boxes. So the first thing we want, remember, is our means. So make note of that. 56%. You can round to 2 decimal places. 56% in the incubated condition, that's A. 47% in the non-incubated condition. So don't worry about this mean difference here. Just take down these 2 values. and the t-value, okay? That's all we're going to worry about for today. So write down these two and this t-value. Next thing, last thing we want actually is going to be our p-value. So remember your p-value, basically what are the odds we got these results just due to chance. And as you can see, quite low. We'll use the two-tailed value for now. P-value is 0.01. Remember the cutoff is usually 0.05. So since it's lower than 0.05, that means that very unlikely to have gotten these results due to chance. More likely it's that our manipulation had an effect. So write down that two-tailed p-value. Okay. So again, that's it. Our manipulation seemed to work. Incubation does seem to help you solve those trials. Okay. So that's about all you need for your lab report that wasn't covered in lecture. So I will upload that in just a minute, and then you can pull that out. And it's pretty straightforward. Nothing too hard in terms of the questions. I mean, we just did the analysis together, so it's not exactly completion. Like you can lose marks on it, but should be fine as long as you pay attention and stuff. Okay.